For this video, we're going to start talking about hormones. So how do plants communicate within their own bodies? Now, imagine a plant just out in the environment somewhere. It has a lot of different environmental stimuli that are interacting with it. It's getting pushed around by wind, it's dealing with competitors of other plants, having to understand where to move nutrients, how many nutrients it has in the soil. And then on top of that, it's also being consumed by herbivores all the time. And all these different environmental stimuli, in conjunction with its own physiological needs, means that plants need a way to communicate within their own bodies. Now, this is very similar to what we do, right? When I move my arms around, I have a nervous system that's signaling for me to move my body around. I'm picking up different stimuli in the environment, and our bodies have both hormonal signals and neuron signals, so nervous system. Plants don't have so much of a nervous system. They do have some electrical impulses that are sent, but it's not really analogous to our nervous system in any way. Their primary form of communication is through hormone signals, just like we have, but that's their primary mechanism. So when we're talking about hormones, we're really talking about plant signaling. How do plants send a signal from one part of their body to another part of their body? And a hormone is really just a chemical messenger, some chemical that has some by default message encoded into it based on the chemical reactions that it'll carry out. So in plants, hormones can do basically everything that the plant does. It can influence which genes are expressed. So it can control which part of the DNA is being expressed at any given time to create or reduce the amount of proteins that are being made. It can influence transcription, so which mRNA is being read out from the DNA. It can influence cell division, increasing it or decreasing the amount of cell division that's going on. It can also influence growth, how fast or slow the plant is going to grow. And we're going to be going through a variety of different hormones and talking about some of their functions. So the major plant hormones, there's a lot of different plant hormones. There's three main groups of hormones we're going to talk about, and then two specific hormones. So the biggest one is auxins. So we'll be talking mostly about auxins and their role in plant growth. We'll also be talking about cytokinins, gibberellins, abscisic acid, and ethylene. So for auxins, we're going to go into most detail about auxins for sure, and we're going to kind of go over it in different steps. So as an overview, auxin means in Greek just to grow. So auxins, our primary role is regulating growth of the plants. Now, very interestingly, and this will become really important later on, is that auxin is synthesized in the apical meristem. So as the plant is growing up, the apical meristem is growing and extending out, elongating, like we've learned in previous videos. That apical meristem is going to synthesize the auxin hormone, right? And so inside tissues that are growing and developing is where auxin is going to be produced, and that's going to modify which part of the plants are going to be demonstrating rapid growth. Now, there's a bunch of different auxins. IAA is really the only physiologically active auxin that's natural, but there's a whole bunch of other auxins out there. Any rooting hormone powders that you have are going to be auxin based. It's a really, really dominant class of hormones. Now, when we're talking about how auxin gets transported around, there's several different mechanisms. We're going to be talking about polar and nonpolar transport, mostly. So here's a little plant. We have roots, and we have a shoot system up here. Now, IAA, that's going to be indole acetic acid. That's going to be the auxin that we're talking about. So polar transport is going to be unidirectional transport based on the cellular transport between cells. Nonpolar transport is really just simple diffusion of auxins. So nonpolar transport can kind of move in any direction based on concentration gradients. Polar transport is directional, and we'll talk about the physiology as to why that is. So we're going to start off with auxin's polar transport mechanism. So let's say we have a plant stem or some sort of tissue here, and auxin is going to be transported down. The plant wants to move auxin down that stem there. Imagine it's being synthesized in the apical meristem, and it wants to send auxin down the stem, maybe to regulate some sort of tropism, right? We talked about tropism before. So maybe this is on the shaded side of a stem, and it's going to bend the plant towards the sun. And so auxin is going to be transported from the shoot apical meristem down the plant. Well, there's several things that are going to happen. We're going to talk about it both in. I'm going to go over this diagram first, and then go over the step by step of it in words. So we have 3 different cells here, right? And they're all kind of lined up. Now here we have these purple ones are called PIN transporters. So they're the protein transporters that are going to allow auxin to leave a cell. Now, if you remember, the plasma membrane is composed of a phospholipid bilayer with a hydrophilic and a hydrophobic portion of it. Because of that setup, auxin that is charged can't diffuse through the cell membrane. Remember, nonpolar molecules, really small polar molecules sometimes can diffuse through, but really charged molecules and really big ones can't diffuse through the cell membrane. So because auxin, we'll talk about why in a second, is charged inside the cell, it exists as an ion, it's negative here, it can't diffuse through the cell membrane. And so it's going to move through these protein channels when they're escaping a cell. Now, what's really important is that the apoplast, the area outside of the cell, is acidic, pH of about 5, and the cytoplasm has a pH of about 7, so it's a neutral pH. That's going to change how auxin behaves. So let's walk through it step by step here. We have a cell up here, and auxin is getting transported through the PIN protein into the apoplast, where it's pH 5. Now, in this environment, it'll survive as AAAH, meaning it has that hydrogen bonded onto there. That means it is nonpolar because it doesn't have a charge there. Now, because it doesn't have a charge, it can diffuse through the cell membrane into the adjacent cell, or it can move through some sort of influx protein. So some sort of protein channel that will allow it to diffuse through. So from the apoplast to the cytoplasm, because it's nonpolar at this point, it can take either channel, right? So it can go through either way. It just enters that cell that's next to it. Now, once it gets into the cytoplasm, where it's pH 7, IAH, with the hydrogen attached to it, is going to dissociate. So it's going to become IAA minus and a proton H plus. So they're going to dissociate, right? So now we have an IAA that's negative and an H plus, a proton there that's positive. Now remember, because it's negative, it can't diffuse through the cell membrane. So if it can't diffuse through the cell membrane, it has to move through a protein channel. That's what the PIN transporter is going to do. So the PIN transporter there will allow it to move through the cell membrane. It'll act as the doorway for that auxin there to move out of the cytoplasm back into the apoplast. And then the same thing happens down a chain of cells. Now, the reason why this is polar transport, meaning it's directional, is because these PIN proteins then dictate which way from the cell that the auxin can actually exit. You can see here there's only these purple proteins on the bottom of the cells. So auxin will diffuse out of the cell and then go down and diffuse into this cell. Move through the protein, diffuse into this cell. Now, theoretically, right, the auxin could just move around, diffuse, and diffuse back into its cell. But once it does that, it's trapped again and has to exit out the bottom. Because of this positioning of the PIN proteins, it means that auxin can only travel in one direction. Whatever direction those PIN proteins are will dictate which way the auxin is going to travel. And so then the plant can control which way auxin will move in any given pathway. So polar transport is determined by where in the cell the PIN transport proteins are, because this polar auxin molecule can't diffuse to the cell membrane, can only use those channels. So let's put that in words for you here. So key traits again, so this is just reiterating exactly what I just said. Cytoplasm is pH 7, apoplast is pH 5, so acidic versus neutral. In the acidic environment, it'll exist as a nonpolar IAAH, and in the cytoplasm, pH neutral, it'll drop the proton and become a polar IAA minus. Now, IAH is going to enter the apoplast, or it's in the apoplast, right? And it can diffuse through the cell membrane or through a protein channel. So because it's nonpolar, it can move right into the cell, no problem. Because there's a higher pH in the cytoplasm, it's going to dissociate from that proton and become an anion, an IAA minus. And because it's charged now, it can no longer move through the cytoplasm, so it must exit through the PIN proteins. And the location of the PIN proteins then dictates which way the auxin will exit the cell. Since oxygen can only exit the cell to dependent proteins, it'll enter the apoplasm on that side of the cell and then diffuse into the adjacent cell in that same direction, creating a net movement as it cycles through this pattern from cell to cell of polar transport. Okay, so make sure to review that material. I know it's a little bit complicated, but just spend a couple minutes working on it and you should be able to grasp it. And definitely come talk to me if you have any questions. Now, oxygen nonpolar transport is much easier. Nonpolar transport just means it's going to move around with the assimilate. So it's going to move either with the xylem up, or it's going to move into the phloem with the assimilate and diffuse out wherever it goes into the cells. So oxygen nonpolar transport is the same as nutrient transport we've talked about before. It's just going to enter the assimilate and then go from sources to sinks. This also means it's not that it's necessarily non-directional, but it's not a dictated direction. It'll just move wherever it's needed. In polar transport, it can only go one way. So nonpolar transport is very similar to what we talked about with phloem movement. Now, oxygen has several actions that are really important. Starting off with how they interact with stems and shoots. So oxygen will change its behavior if it's in a shoot or in a root. And you can kind of see that here. So where's my cursor at? Percent stimulation means how much is it going to grow. And percent inhibition is how much it's going to inhibit it. Now, you can see as concentration of oxygens increase, root inhibition increases. So oxygen will inhibit root growth. Now, up into very high concentrations, it stimulates stem growth. Now, at very high concentrations, oxygen is an herbicide, so it will kill all plants at very high concentrations. And so that's why you see this drop off here. But in general, oxygen stimulates stems and inhibit roots. Now that means depending on where oxygen concentrations lie, it can change the tropism of the plant and where it's going to grow. So let's look at this, for example. So we've looked at this before. We have a stem shoot there. You cut off a little piece of it. If you do nothing, it grows back up, grows through the tip and grows up. If you put the tip back on, it'll curve over. Or if you add oxygen to it, it'll move over. Now remember, oxygen will stimulate root growth, and it goes down in a phototropism situation. Oxygen goes from the shoot apical meristem down the shaded side, causes cell elongation and division, and causes it to bend over towards the sun. Now, that's oxygen stimulating stem growth. Now, it can also trigger root formation. So, specifically adventitious roots. So, you have a propagate, you cut it off, right? You add oxygen to the base, and it'll form roots. This is pretty critical to how propagates work, right? Behind me in various places, I have some propagations here. And these are just cut off pieces of plants that I have put inside of oxygen, and now they are rooting. So let's see if I can pull out. This is a pothos. Pothos roots pretty well. You can see these are all roots here now. And that's all triggered by the synthetic oxygen, IBA, instead of IAA. If I can get that back in there here. Now it doesn't want to go back in the jar. Pothos is known for producing a lot of rooting hormones, so I often keep it inside with other plants. So that propagation technique that we use for a lot of agricultural and horticultural plants depends on that oxygen. It depends on that rooting hormone, that oxygen allowing root formation from a non-root structure, like a cut-off stem. Now, it also prevents abscission. So abscission is the dropping of various body parts, usually leaves. So leaves will drop off in the winter if it's a deciduous plant, and oxygen acts to inhibit that. So oxygen will block another hormone called ethylene. Ethylene we'll talk about in a little bit, but it's the hormone that can cause problems with fruits in your refrigerator or on your counter. Because if you leave bananas in a bag or something like that, or bananas with another fruit, they'll produce a lot of ethylene and cause it to ripen up and eventually rot really fast. So ethylene also is responsible for senescence and abscission, for the dying of structures and the dropping of structures of the plant. So oxygen blocks ethylene. So oxygen therefore prevents abscission of leaves and different structures. So if oxygen concentrations are high, it won't go through abscission. It won't drop its leaves and go through the senescence process. During the season, as auxin levels begin to decrease and ethylene increases, it'll go through the seasonal changes associated with senescence and abscission. Now you can kind of see that here. So they have some plants here, right? And they cut off a stem, but they put on a paste of it containing auxin, and one of them that doesn't. Now you can see here, this one maintained its little petiole there, and this one drops the petiole. That's because the auxin they put on this cut stem prevents it from being abscissed. It's not going to allow it to fall off. But since they didn't put auxin on this stem, ethylene took over and did allow it to absciss that broken petiole. And so auxin will prevent that abscission from occurring. Now, auxin also promotes fruiting, and it also does something which we call maintaining apical dominance. So here's a fruit, here's a strawberry, right? Now imagine you took all the seeds from the fruit. If you didn't do anything else to it, the strawberry would basically sit like that. Without the hormones from those seeds and the developing seeds, it's not going to ripen into a full fruit. But if you take that, after you remove the seeds, and if you add auxin to it, it will still create a strawberry, but just seedless. This is one of the tricks that a lot of agriculture will use to develop fruits with seedless varieties. They will just add auxin to it in order to stimulate early mature fruiting of the plant. Now, apical dominance you can see over here. So here's a little plant. You have a terminal shoot here, right? If you cut off that terminal shoot, that shoot apical meristem basically, you cut that off, it will send out a lateral shoot. That axillary bud will mature and start to grow out. This is one of the tricks a lot of farmers will use. If you have tomato plants, rather than letting them grow really tall and get kind of leggy and gross looking, if you want to increase the amount of fruit that they're going to make, cut off the apical meristem when they're maybe about this tall. Maybe if they have 3 or 4 different branches, you cut off the apical meristem, and the axillary buds will mature. So the plant will stay shorter, but it'll grow bushier. That is because when you cut off the shoot apical meristem, the auxin that's produced there is no longer around. So the auxin from the apical meristem gets removed, and therefore auxin reduces. And since auxin promotes apical dominance meaning growing from the tip being the dominant form of growth, that apical dominance gets broken, and the plant will now grow laterally. It'll grow through not secondary growth, but axillary bud growth. Now, if you do the same thing, cut off the shoot apical meristem, but then put some auxin on the tip of that cut piece, the terminal buds still stay dormant. So the axillary buds don't start to grow. Rather, they just stay dormant because that auxin is still inhibiting their growth. So auxin maintains apical dominance in the plant by suppressing axillary bud maturity. Oops. Now, auxin is used for all kinds of things, like I've talked about already with the plants behind me, for propagation. So IBA, indole butyric acid, I believe, it will trigger. It's a synthetic way to trigger rooting in plants. That's what's inside this rooting hormone here. It also triggers fruiting, so it can trigger fruiting and early maturity of fruits. It'll also reduce fruit drop. Remember, it reduces abscission, and a lot of plants will drop their fruits when they're mature, and it'll stop that from occurring because it blocks that ethylene hormone. Now, in really high concentrations, auxin is a herbicide. And so what likely happens is it probably causes an issue with polar transport when you add too much auxin to it. Now, this is particularly useful for dicots. Auxin herbicides work really well on dicots, not so well on monocots. And so it kind of varies a little bit there. Okay, now we're going to cytokinins. Cytokinins should sound to you like cytokinesis, the separation of the two cells at the latter part of mitosis or meiosis. And cytokinins do that. They promote cell division, and they also promote shoot formation. Now, there's a bunch of different ones. There's over 200 different natural and or synthetic cytokinins, so a very wide variety of different molecules out there for it. They're really helpful during the germination process. So when the seed starts to germinate, it needs to grow very rapidly. To grow really rapidly, you have to have a lot of cell division occurring, and then therefore you need a lot of cytokinins around. Now, it also often opposes auxins. So cytokinins will start to act against apical dominance, and it'll also inhibit lateral roots. So auxin wants to produce rooting or promote rooting, and promote apical dominance, and cytokinins will do the opposite. And so the relative ratios of those will determine how the plant actually ends up operating. Now, the way cytokinins typically work is they just influence gene expression. So they're going to go into the cell and modulate which genes are being expressed at any given time. Next, we have gibberellins. They're really responsible for cell elongation. So making cells elongate really quickly, which allows plants to grow very rapidly. There's a lot of these as well, about 125 different versions of gibberellins. They're synthesized in all kinds of growing tissues, so the roots, shoot apical meristem, the young leaves, and seeds. Now, gibberellins are really helpful in seed germination. So when water gets taken in to the seed, right, it's going to activate gibberellin. Now, gibberellin will then activate what we call amylase. Amylase is an enzyme. It's the same enzyme that we have in our mouth. So if you hold some food in your mouth or something like that, or some bread in your mouth, it'll start to dissolve and it'll start to taste sweet. That's the amylase breaking down the carbohydrates. In this case, amylase is breaking down the starch inside the nutrients, or starch inside the seed that's held inside the endosperm, making it into glucose that the plant can then use for cellular respiration as an energy source. So gibberellin is responsible for activating the enzyme that allows the endosperm to function as a food source. Now, because it's also responsible for cell elongation, it's also helped with shoot elongation. So rapid division elongation of shoots and tendril formation. So the tendrils growing out of the tips of the shoots, allowing them to wrap around different plants. Now you can see here in this plant, it has a little basal rosette here, and then this really long stem. That's called bolting. A lot of plants will shoot up a stem really quickly, really high up as they start to flower, and gibberellins help with that bolting process. It also helps with breaking dormancy, like we talked about here. As water goes into the cell, it's going to activate gibberellin, and helpful for fruit production because it's rapid elongation and growth of the cells. So here's a little summary table I thought was useful for those main hormones we just talked about, the auxins, cytokinins, gibberellins. So if we start here on the top with the cytokinins in yellow, gibberellins in green, and auxins in blue. So cytokinins slow down the aging process of the plant, while gibberellins increase cell division, and cytokinins then also trigger opening of the help with stimulating the opening of the stomata in leaves. Auxins formation of the leaf blade. Gibberellins interrupting plant dormancy. Yellow cytokinins stimulation of cell division. Now when we talk about roots over here, this is mostly auxin, formation of root buds, stimulating lateral roots and lateral root formation. So auxins are really prominent in roots there. Now when we're talking about lateral buds, so stimulation of lateral buds by cytokinins, induction of germination by gibberellins, stimulation of cell division by auxins. And you kind of see how they start to interplay with each other there. But there are other hormones. One of them you've already heard about when we talk about stomata regulation, the hormone abscisic acid, or ABA. It's a plant growth inhibitor, so it's going to reduce the growth of plants. So the abscisic acid should sound like the word abscission to you, like we've been talking about, but it's actually a misnomer. They used to think it triggered abscission, but we now know that's not true, but it still has that name. What it does is it inhibits division and expansion of the cells. So the other ones we talked about typically are encouraging growth, with the exception of apical dominance by auxin. Abscisic acid is doing the opposite. So ABA will accumulate during times of stress. So when the plant starts to get stressed out from a variety of environmental factors, it's going to help trigger that stress response. That could be closing up the stomata, like you would want in dehydration. It's also going to help regulate seed dormancy. So ABA is going to be present inside of the seed, and over time it slowly breaks down and deteriorates. Once it gets low enough, it's no longer inhibiting the growth of the embryo. And so in that way, it's a regulator of seed dormancy by being an inhibitor of growth. It also helps with bud dormancy, so it stops the growth in the axillary buds. And so if there's a lot of abscisic acid there, the axillary buds aren't going to grow. We've already talked about how it triggers stomatal closure. And then there's a lot of, as with all physiology, there's a lot of interactions with other substances. Now, ethylene is a little bit different. It's a gas. It's a volatile gas that doesn't mix well with water. And so As ethylene gets created, it also escapes really quickly. And so plants will be releasing ethylene gas in their bodies quite readily. Now, when ethylene gets produced, it's doing it from usually maturing tissues that are wilting, that are ripening, as well as some nodes and different things. But as the plant matures, those mature tissues or dying tissues will produce ethylene. Now, ethylene will create or cause senescence, so it causes aging, so it causes maturation of the plant. It triggers leaf and fruit drop, so causing the leaves to fall off or causes the fruit to fall off. So typically, if you're a farmer, you're not usually a big fan of ethylene. It can cause your fruit to ripen and rot really quickly, cause your trees to drop their fruit and get damaged. So typically, you don't want a lot of ethylene around. It's also responsible for what's called the triple response, basically how plants get out from under debris. So ethylene causes seedlings to grow really short and really wide, but also causes them to bend. And what that means is as they grow, if there's little debris, say like mulch, they can get their way out of there. Now, if you do want to artificially ripen a fruit, adding ethylene would do that. And so ethylene is often added to ripen fruit artificially. But then also in fruit storage containers, you don't want ethylene around if you don't want your fruit to ripen. And so you often need to remove ethylene from the air in order to stop that from occurring. So here's a little summary table of that. This is just for your review. Just reviews what we just talked about for those different hormones. Remember, auxin, cytokinin, and gibberellins, those are all groups of hormones. Ethylene and abscisic acid are specific molecules. There are other important hormones and different signaling molecules as well. So you got the brasinosteroids. This is a really diverse group. A lot of times they act in conjunction with other hormones, but they can be beneficial in elongation, division, growth, germination, and almost every other piece of the plant they play some role in. You also have cysteamine. This triggers jasmonic acid if something starts to eat a plant. So say you're a little plant and you have some sort of an aphid or something starting to chew on your tissues, right? Well, that aphid is hurting you, and you want to get rid of those aphids as quickly as possible. So that herbivory will trigger cysteamine. Cysteamine will then trigger jasminates. So down here we have jasminates that are going to be produced when the plant is starting to be consumed. So what it will do is it will trigger a variety of proteins to be created and different enzymes and different things that will, one, make the plant taste not so great, and so maybe the herbivore doesn't want to eat it. But it also releases a protease inhibitor. So proteases are enzymes that break down proteins. And if you're inhibiting those, it means you're producing a molecule that will inhibit the breakdown of proteins. Now, what they're trying to do there is basically make it to where protein digestion for whatever herbivore is eating it becomes difficult. And that can cause digestive issues, maybe even death in some organisms. And so the goal is to make it to where the plant's less digestible and the herbivore will leave. It can also, in some ways, attract other predators for the pests. So, like the enemy of your enemy is your friend. The plant, say you have an aphid, you might want to call in some ladybugs or some other things in there to come in here and eat those aphids. And so you're trying to attract the predator of your pest, and some jasminates may play that role. We also have salicylic acid. This is a molecule that is essentially aspirin. So it's produced really, really commonly by the Populus group, so by aspens. And it's produced a lot of times in the bark. And so it is a way to have an immune response to some sort of parasite, herbivore, predator kind of infection, right? So it can be toxic. So it can also attract predators. There's also some evidence to show in some plants it helps with increasing metabolic heat production. So when it's trying to break through the snow, it can melt the snow around it and it can sprout up through it. And then the last one we're going to talk about is phloragen. This is one that's going to use the day length cues, so the phytochrome R and phytochrome far red. And when it's appropriate, it's going to be produced to trigger flowering. So phloragen is a signaling molecule that's helpful for initiating the flowering process. Okay, so to recap, we've talked about a variety of plant hormones, including auxins, cytokinins, gibberellins, ethylene, abscisic acid, brasinosteroids, cysteamine, jasminates, salicylic acid, and phloragen. Make sure to you pay a lot of attention to those, especially the main ones, so auxin, cytokinin, gibberellin, ethylene, and abscisic acid. with a lot of attention towards auxin. And then definitely come talk to me if you have any questions.